Welcome everyone to our virtual presentation for this case study. So right now we are going to present about data cleaning in very retrospective related to clinical analysis and clinical treatments. So uh, we're going to cover a lot of things which are related to clinical analysis, like how to figure out what to do and how to go about it, like where to get the, like how when once you get the data, what to do about it, like how to perform cleaning, uh, pre-processing, all kind of diagnostic analysis, error analysis. And this case study is really about documenting all these things. So we're not going to deep dive into code and all that. We're just going to cover like what are the basic steps which should be considered and should be taken in a case study which deals with clinical data. So just go back. So we're going to cover introduction, like what this whole thing is about, a brief history of data cleaning, screening phase, and a couple of phases are there, diagnostic, treatment, then cleaning in general, and with retrospective to clinical data, and documentation and reporting that is, represented as the guidelines for these kind of analysis. So moving on, so you might have heard this quote that move fast and break things, startups, California, Silicon Valley. But in terms of clinical AI, it means killing people, because if you try to do that, uh, many startups have done that, and they, eventually didn't turn out to be good. And there are a lot of things which should be ethically taken care of, especially in clinical AI, because the results, even a significance of 0.5 difference, 0.5% difference can change outcomes. So moving on. So basis of our research is one of the paper that was published in around 2005. This paper's primary author was Van der Moik, and along with there a couple of other authors as well. And this paper, like before paper, like this paper, there were not necessarily any guidelines or any kind of, a coverage on all the clinical studies that are being carried upon, and what are the guidelines, what are the, principles that data visualization, cleaning, and everything should stand off. So there are a series of these papers that these authors published. This is one of the paper which briefly talks about more, in the cleaning area, like what to report, what not to report, how to get started with your data. And there's other couple of papers which, followed this after the success of this, that talks about visualization and all kind of modeling processes. So a nice fact on this, that in general machine learning, if we just, visualize, like we are just visualizing the data, if we look at the data, we get an understanding that there are some outliers and we just go and ignore them, or maybe just drop them, or, just we don't focus that much on the outliers. But it turns out in clinical setting, it was found, through this paper and the researchers as well, that considering outliers does increase the coverage of the model, and it gives better results when you actually test those models out in, the public, because there are all kind of varieties of uh, a data collection that was not performed because it cannot be performed, you cannot sample the population. So it's a general thumb rule. uh, this paper also like, highlights this fact that outlier detection is quite important, and you can't simply drop them or ignore them. As a fact, cancer detection, one of the primary areas of clinical AI research, has a more success rate with outliers in the data as compared to, without the outliers in the modeling process. So this study provides a kind of a framework for, uh, showing everything about the principles of different kinds of error types, if you're reporting or not reporting, what kind of report you should make. And it also serves as a, prerequisite if you're going to in a clinical setting. It serves as a prerequisite that what things you should take care of while performing data cleaning and pre-processing. So a brief history also, early 2000s saw a lot of trial errors. So like people were having trial errors in the reports as well, but usually what was happening in the documentation, it was missing. So if you read a documentation from a report, early 2000 or before that, you won't see a lot of chapters or a lot of discussion on what kind of clinical trials were conducted, what kinds of errors were reported, because these are not documented. These were like just ignored simply. And that turned out to be uh, quite a ignorance in the setting, because it was not detecting the models, and the procedure that was being followed was not uh, fitting on it. It was fitting the theoretical values but not on the actual people. So a lot of things were missing from the visualization and cleaning area in the reports. Then 2010s saw again, like, the data cleaning still was a suspect activity. It was not the major performance changer for a modeling process in a clinical setting. So most of the studies still ignored. Uh, they covered the clinical trials now, like from 2000 to 2010s. They started reporting clinical trials that they conducted this research based on this many sample of people, but still they were missing out on the data editing part, reporting the errors, and all these things. So, flashback to these parts. And then now in present 20s to around 20s, most of the studies are covering all these topics. They're reporting clinical trials, they're covering coverage, they're covering all kind of, comparison between models with and without outliers. And now, as it turns out, for the clinical as well as in the general setting, data pre-processing, cleaning, takes about 80 to 90% of the time, while just 10% for the modeling and documentation and everything else. So yeah. So moving on, I'll hand it over to Jonathan.
Thanks, Pranjal. So the first step in the clinical data cleaning process is the screening phase. The goal of this phase is to identify 4 different types of data oddities. These oddities are lack or excess of data, outliers, strange patterns in some of our distributions, or unexpected analysis results. There are a few methods for detecting these data oddities. So we have sort of 2 camps, the exploratory methods and the statistical, more mathematical methods. Some of these exploratory methods include validating data entry and double entry. So this would be both in the data input stage and also the data collection phase, where, for example, when you go online to create an account somewhere, they ask for your email and a re-enter of your email to make sure that you're putting in correctly. And then we have the browsing of data tables after sorting. So you can sort by in ascending or descending order and sort of check manually to see if you have any erroneous inputs. We can also print out variables that don't pass range checks. So if you know your data well and you know what the values can't be, let's say they can't be negative or they can't be above 100, you can filter by these ranges and see which variables aren't passing. And also we have graphical exploration. So scatter plots, box plots, any sort of plots you can think of, pair plots, to sort of explore how your data interacts with each other and see which variables are in the right place. And then we have some statistical methods. So frequency distributions, summary statistics, those are pretty standard. Z-scores and IQR are really helpful for detecting outliers. And IQR can also be done visually or exploratory with box plots. One thing to note here is that there's also erroneous inliers. These are data points that are generated by error, but they fall within the expected range of our data. So you can see in the image on the right there, the orange data point is an erroneous inlier. It's within the expected range of X and Y, but it's still not right and shouldn't be there, could mess up your analysis. And in green there, we have an outlier. This falls outside of the range of X and Y of most of our data points. These inliers can be tricky, these erroneous inliers. They're sort of hard to detect, and there are a few methods, but there's not a clear-cut way to sort of view them. You can do relational plots like scatter plots and line plots. You can also do some regression analysis, Or you can do remeasuring of data points if that is possible, sort of double checking that your inputs are correct. And I'll pass it off to Shania for the next phase.
Thanks, Jonathan. The next phase in analyzing clinical data for errors and, as Jonathan talked about, inliers, outliers. The purpose of the diagnostic phase is to truly find the true nature of worrisome data points and patterns and statistics inside the data that you have. So Jonathan explained the erroneous inliers and some outliers. When we look at data, there are four ranges in which clinical data can fall in. It can be normal, and normal data can comprise true normal data, which is true data and the data that we want to analyze. And it can also contain erroneous inliers. There's suspect data, which falls in a range above normal, but not in a range where it can be considered a complete outlier. So it can contain erroneous outliers and idiopathic, non-explainable data. And then there are some impossible values that just cannot be, for example, age more than 200 for a human being. So the impossible data range can contain erroneous outliers, idiopathic, and true extremes. And then there's the fourth type of data, which is missing data. That data just wasn't collected during the collection process, or it was a survey and it wasn't filled out by the user. So that could comprise of the missing data. So what do we do in the diagnostic phase, and how can we go back to these values and find out what was it, an error, or is it a true extreme? So there are 4 ways explained in the paper that you can use to kind of deal with these outliers and these strange values. First one is to go back and look at the tests and see if that particular value is consistently the same. Is it consistently in that range, or was this one particular data point just in the wrong range? Then there are methods where you can use features either inside the data or outside the data to confirm your value. So for example, you could use, if there's a very strange body fat percentage number for a person of a certain height, you could use their weight to kind of confirm your data point, if it's actually a strange value or is that a true value. Another way in clinical data is to kind of go back to the users and the participants you got the data from and follow up with them, ask them more questions that can confirm the data point or get information on if it's correct or not, or ask follow-up questions that could provide you more information. And then fourth is using common sense and the domain knowledge that you have in the field that you're working to kind of get a sense of the true nature of the data. And mind it that these procedures can only be used and these can only work if data cleaning happens at a very early stage in the data collection process. And sometimes it's not possible to go back to the participants. Sometimes it's not possible to remeasure certain values. So it's very important to start thinking about data cleaning as soon as you start collecting data.
All right, so like in medicine, once we've screened, once we've diagnosed, now it's time to treat. What are our treatment options? Well, once you've identified the errors, the missing values, and our true values, whether they're extreme or normal, we really have 3 options in our data science tool bag. Either we can correct the data, we can delete the data, or we can just leave it unchanged. Now, with correcting the data, the context of this paper was a medical data. So when the correct data is finding the correct value. In our cases, we're not selecting the data. We aren't always involved in that process. So correcting the data means substituting with a mean, doing some correlation analysis, something of that sort to get a value in there that can fit with the type of analysis we want to do. We can also delete these values. Now, sometimes if we have impossible value. Say we have a height measurement and that measurement is 0. Well, that's an impossible value. That's going to mess up our analysis. And as we know, we're treating our numeric data as vectors. So that will really affect how our models are going to predict what we want it to. So it'd probably be best to just delete those values. But if you can correct it in the way that does fit with the model that we're trying to work, then that should be your first option. But oftentimes we just delete. And then finally, we can just leave them unchanged. Now, usually we don't leave them unchanged, especially with impossible values, as I mentioned, but sometimes we're going to see some variation in our data, right? Not all measurements are going to follow, say, a strict linear regression. There's going to vary a little bit. So we have to account for that variation when we're considering what kind of methods we want to employ. And like I said, in the context of medical research, some researchers have noticed that if you have, say, temporal data and two observations that were measured very quickly one after the other, you can just take the average measurements between those two, and they've actually found a boost in accuracy between those. So like I said, oftentimes we correct, we delete, but there are going to be some situations where we don't want to do anything. So how do we know when we're treating or when we aren't supposed to be treating? Some researchers have found that, or suggest that, you shouldn't be altering these extreme values. Like I said, there is going to be some variation, but we don't want to take all the variation out of it. It's not going to be a very good model that way, because we're not going to see data with no variation in the real world. We want a model that can be generalized to new data that we collect that we don't already have. So this 1994 textbook by Gardner and Altman suggests that you should keep extreme values in the analysis. But in practice, that's not always going to happen. We're going to want to change our data. Usually, sometimes we normalize it. Sometimes we just fill in missing values. But sometimes what we see is not just normal variation. Sometimes it's just errors in the data collection. As we know, data is collected by humans, so it's not going to be perfect, right? So as a data scientist, you just have to use your best discretion when you're treating these, these worrisome data points. And, oh, sorry, to go back. So data cleaning is a really important process. And in the context of this paper, often if you are closely related to the people collecting the data, then you can use what you've done in the data cleaning process, the treatments you use to sort of inform how the data should be collected in the future, or how the new data need to be collected to affect your study.
I'll continue to explain the key differences and elements of data cleaning in general and for a study-specific process. As mentioned in the paper, data cleaning in general is allocated a hefty portion of time in real-life data science projects. And in majority of the situations, irregularities and inconsistencies are discovered incidentally. According to the authors, it is ideal to have a predefined set of protocols that guide the data cleaning process. And this predefined set of actions are characterized into screening, diagnostic, and editing, as explained earlier by my teammates. Following a preset of rules for cleaning and cleaning, it conserves the notion of prioritization, which is essential if we have a time constraint. It is mentioned that errors in particular variables such as date and gender are important to address earlier because they induce significant impact on the analysis. For example, if there are errors in date variable, it can affect the temporal analysis of the data, and similarly, errors in gender can impact the demographic-based analysis, which can lead to bias. One approach to handling errors is identify and correct them through data cleaning and validation techniques such as cross-validation and outlier detection and imputation. Coming to the next slide, in terms of data cleaning as a study-specific process, the foundations of the aforementioned elements still exist. The difference is how the approaches are curated to benefit the study in terms of time and efficiency and so on. The choice of error detection methodology should be tailored to the specific requirements and characteristics of the study. study data to ensure accuracy and reliability of the analysis. Apart from the general intuitions on how to proceed with the cleaning, the authors have mentioned scenarios where inconsistencies have different sort of effects in different scenarios. In small studies, the impact of single erroneous data point can be significant and have a distortion effects on the analysis. This is because the sample size is small and the outliers have a greater effect on the data that we have. In larger studies, it is important to check the temporal consistency of the data to ensure that the measurements and observations are consistent over time, such that checking if a person weight is recorded consistently across all the time points, and inconsistency measurements can be effectively validated and reliability of the analysis. Additionally, in the study-specific data cleaning, independent expert examination is needed to avoid induced bias by using his domain knowledge, as mentioned earlier. Yeah, that's about it. These are the key differences between a data cleaning as a generic process and a study-specific process.
So coming to documentation reporting. So now I'll talk about the importance of documentation and reporting in data cleaning phase of clinical studies. So the author of the article mainly talks about suggesting a data cleaning plan. So every research needs to include a data cleaning plan, which kind of includes the details about what's the budget, the personal requirements, what are the screening tools used, what are the diagnostic procedures mentioned, what are the treatment methods used, what are the decision rules, and everything. Next, please. And mainly in case of like clinical studies, there will be committees like monitoring the data and the safety. So these committees need to get a detailed report regarding the data cleaning methods. And this committee will kind of give a feedback to the ethics committee. So in all of this process, it is important to maintain the transparency in data cleaning. So in the medical reports that we submit to the committees, there need to be information regarding the error types, what are the error rates, what are the deletion and correction rates that you've used, and how did you impute the values into your data sets, and why did you impute them? I mean, how they kind of affect the results, how the results would vary with outliers and without outliers, and what are the kind of all the data cleaning methods that you've used in the overall procedure. And next, please. Let's kind of understand the importance of data documentation and reporting using a case study. So let's take a study where they're kind of testing the safety and efficacy of a new drug. So when it comes to testing a drug or safety, there needs to be information regarding the drug and everything. So coming to compliance, the data cleaning process should kind of comply. The process should comply with good clinical practice guidelines. So these are the guidelines that are that need to be followed when conducting any case study. Coming to safety concerns, so if we use any unreliable data or incorrect data, the results could be varied. So I mean, it could harm the patients if the results are not correct. And next, coming to reproducibility, if the data cleaning process is not properly documented or there are any errors, the results may be inaccurate, and this may lead to difficulty in reproducing the results. And coming to traceability, if the results are not properly documented or there are any errors in the data, then it would be difficult to trace the exact results again. And communication, and if there's no proper communication, it would be difficult for the stakeholders and other people in the whole process to kind of understand what's going on in the research.
All right, that was our presentation. Thank you for watching.